Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Sunday, January 5th, 2014. Our first story of the new year is exciting news from the world of material science. A team from Berkeley Lab has been working with an interesting material for the creation of microscale motors and actuators. In case you didn't know, an actuator is essentially an artificial muscle. Instead of using energy to generate rotation like a motor, it contracts or stretches in one direction. But for both of these purposes, it seems that the material vanadium dioxide is perfectly suited to the job. It is already a rising star in the world of material science because it has some unique properties. For instance, at low temperatures, it acts as an electrical insulator, but above 67 Celsius, it becomes conductive like a metal. This kind of change in property due to temperature is called a phase transition, the same as other materials becoming solids, liquids, or gases at certain temperatures. Vanadium dioxide not only has a change in its electrical properties at a certain temperature, but its physical structure as well, usually contracting in one dimension while expanding in two others. So the team demonstrated its actuating potential by creating a tiny metal coil containing vanadium dioxide and chromium. When some electricity was passed through the coil, the chromium generated heat, triggering the phase change in the vanadium dioxide. This small actuator was able to launch something 50 times its weight, a distance of 5 times its own length, in about 60 milliseconds. To put that in perspective, that is about 50 times more powerful than a human muscle of the same size. It is also the most powerful and efficient artificial actuator currently being investigated. There are a wide variety of microelectronic and micromechanical systems these actuators could be used in, and this team is just now beginning to explore their potential. Next is an update from the world of evolution. Researchers from Harvard have been studying a potential mechanism for rapid evolution in organisms with intriguing results. Evolution is generally a very slow process, especially with complex organisms like fish, which this research was mainly based on. Instances of rapid evolution do occur, though, and a gene and its protein product they studied has been shown to relate to a rapid adaptation in a variety of species, including fruit flies and some plants. The idea is known as standing genetic variation, which basically suggests that most populations have a variety of genetic mutations, but that they aren't expressed in the organisms. But if the environment changes rapidly, that stress can essentially trigger those mutations to become expressed, giving a population a major boost in genetic variability and increasing their chance of survival. Or, to put it another way, it's a genetic plan B through Z that gives evolution more to work with when shit hits the fan. This particular gene/protein responsible for the mechanism, at least in some cases, is HSP90. To put this idea to the test, they did a number of experiments using a small species of fish found in a river in Mexico, which also has a subspecies adapted to living in underwater caves. Unlike the normal fish, the cave dwellers have adapted by ditching their pigmentation, getting better sensory organs, and most obviously losing their eyes. Some of the first experiments involved blocking the gene with a drug. Normal fish raised in these conditions showed extensive variability in the size of their eyes. Similarly, cave fish given the drug had variability in the size of their skulls, where their eyes used to be. Even more conclusive, another experiment raised normal fish in a variety of conditions that were more harsh and cave-like, resulting in the same eye variability. This is likely because the physiological stress of the new conditions disrupt with the functioning of this protein, which assists in the folding of other proteins related to gene regulation. More experimentation is needed, but the researchers think this gene is likely responsible for standing genetic variability in many organisms and the key to species rapidly adapting. We end with a quick story from the world of medicine. Mostly quick because it relates to induced pluripotent stem cells, and we have talked about that a lot. But as a quick refresher, iPS cells have extremely similar properties to embryonic stem cells in their ability to divide and differentiate into virtually any cell type. Fortunately, however, they can be created from adult cells using a cocktail of growth factors, sidestepping the ethical issues of embryonic stem cells, while also providing a perfect genetic match when these cells are eventually used in practical medicine. There are still a variety of issues to work out before the stem cells get to that point, though. One of the issues being the speed and efficiency at which adult cells can be converted into stem cells. Currently, it usually only works on a fraction of the cells exposed to the aforementioned growth factors and takes several weeks. But that may be a thing of the past thanks to researchers from Barcelona. This group was previously working with a growth factor to directly convert one type of blood cell into another type of blood cell, known as transdifferentiation. When they added this new growth factor into the usual procedure for creating induced pluripotent stem cells, they found it acted as a catalyst, just speeding up the entire process to take only a few days. That's because its main function is to unravel a section of DNA which happens to have the main genes related to pluripotency, allowing them to be expressed more readily. This encouraging development will hopefully accelerate stem cell research and make it more practical for medical applications in the future. Well, hope you enjoyed this episode. What was your favorite scientific breakthrough of 2013? Let us know your thoughts on that and all the stories in the comments.